Yes, so today we are starting with the second species of Streptococcus, which is Streptococcus pneumococcus. Okay, so according to the first point, uh, the morphology of this, it is the gram positive organism. Okay, gram positive and lanceolate shape organism. Lanceolate shape. So what is the lanceolate shape? It is like this. It has the tapers end, wide end, in the narrow, and from the above it is so narrow. Okay, from the base it is wide. Then it show soluble bile soluble it is bile soluble organism, and the rest of the species are bile insolubility. So it is the only organism which shows bile solubility. Okay, and the second is the optic in sensitive organism while rest species are negative to this test. So this is the basically the differential diagnostic of Streptococcus pneumococcus from the different species. Like we can differentiate Streptococcus pneumococcus from Streptococcus viridans, like it is optic-in positive and it is optic-in negative. Okay, so what is optic-in test? Optic-in sensitivity, in which the laboratory assistant will take this optic-in sensitive plate and a small droplet in the center we put optic-in plate. Okay, so we can see that there is inhibition of growth of this organism in the nearby this area. So we can see that there is no growth of pneumococcus. Okay, so next is the biochemical reaction. So in the biochemical reaction, you can see that this organism is cat positive, which is catalase positive, and oxidase. Sorry, this organism is both cat negative and oxidase negative. Okay, so it don't show these both, and you can differentiate it from the staphylococcus. And the third is, we already discussed, that bile soluble. It is the bile soluble organism. Resistance, if we talk about the resistance, we can see that it is a delicate organism and it kill by boiling in 15 minutes at the 52°C. So you can see that how delicate this organism is. Third important point, antigenic structure. And you can see in the antigenic structure that it creates immune response towards body. So basically what is seen like this, if this is the organism and this is its capsule, it contains antigen C, which is the most important antigen, and by this antigenic structure you can perform a test called Quilling's reaction. In Quilling's reaction, what we have to do is, um, generally this organism create antibody toward this antigen C. Okay, so these antibody which are against this antigen C, we took these antibody antibody plus methylene blue. Okay, when we add these antibody with methylene blue, you can notice in the solution that pneumococcus, this organism is swell. So we basically see that the organism will swell in the Quilling's reaction. Okay, because, and the most important, C reactive protein, you heard about this in the pathology. What is C reactive protein? It is basically first secreted by liver, and it is only the observed, sorry, first observed by the scientist in this organism only. So that's why it is called C reactive protein. C means that it is the antigen C, which is present in the pneumococcus. That's why the name discover from this organism, because the discovery of this antigen C leads to the discovery of C reactive protein. Then if we talk about the toxins, you can see that there are various toxins secreted by this pneumococcus. First is the hemolysin. Example is streptolysin O and S. In my previous video, I told you about that streptolysin OSN, which is heat stable and heat labile. Then second is the leukocidin, and third one is the pneumolysin. Okay, and this organism show alpha hemolysis in blood agar, means it show partial lysis of RBC and show green color. Okay, so this organism show alpha hemolysin. Okay, 6 minutes. Okay, now if we talk about the pathogenesis, in the pathogenesis, oh sorry, I told you, forgot one thing. It also show beta hemolysis. Okay, but when there are certain condition when it show beta hemolysis, the main condition is anaerobic condition, because it is the anaerobic condition is produced by the toxin which is hemolysin basically, which is streptolysin O, and you all know that it not survive in O2, therefore it show beta hemolysis also. Now pathogenesis. In the pathogenesis we can see that the main diseases, that is lower pneumonia, it caused then broncho pneumonia. Okay, then meningitis, then otitis media, inflammation of middle ear, and sinusitis, then peritonitis. These are the main diseases which can cause lower pneumonia. As you all know that if this is the lung, there is the consolidation like property in the whole lobe of the lung of the patient, and in bronchopneumonia the consolidation like property you can see, and it affect also the bronchus part. Okay, so, and next is the last one is treatment. For the treatment you can give penicillin, and if the organism is resistant to it, then you can give cephalosporin. Okay, so this much all. Thank you.